In this class, we shall continue with the next topic, that is the formation of male gametophyte. We have done with the microsporogenesis, which is the process of formation of microspores. So now in this class we shall learn how that microspores develop into a fully matured male gametophyte. So now let's rewind for what we have done. In a pollen sac, there is a microspores mother cells. So microspores mother cell. This microspores mother cells, it undergo meiotic division, resulting into a tetrad. This tetrad got separated. They separated from each other, forming 4 microspores. So now we shall learn how this microspores it develop into a male gametophyte. So this is a microspores. It has a This is a microspores. This microspores has its own nucleus and its own plasma membrane. Plasma membrane. So after a period of time, this microspores develops and develop a layer which is develop a layer around it. I'll draw with a colorful pen so that you'll understand. This layer is known as the intine. It is made up of cellulose and pectin, and then this is the internal layer, and then the external layers of this grows above it, and it grows in an irregular manner. It grows in an irregular manner. It grows in an irregular. There are some portion of this in which the exine does not grow, but it is for a particular reason. So this outer layer, it is known as the exine. This enzyme is made up of sporopollenin, which is resistant organic substances released by a dapitum. So this is a pollen grain. This is a pollen grain. So now we shall draw another one. We'll draw another one here. This is the plasma membrane. This is the intine. And this is the enzyme. This is the enzyme. This pollen grain, it will undergo mitotic division, which involve both karyokinesis and cytokinesis. So firstly, it will undergo a karyokinesis in which this nucleus is divided into two, results into two. So from here, when it has divided, this one is known as the vegetative or vegetative cells, or the tube cells, whereas this is known as the generative cell. This vegetative cells, it has a dense cytoplasm, whereas this generative, it has, after karyokinesis there is cytokinesis. So when it divides, this generative cell is a larger cells with a dense cytoplasm, whereas this generative cells is the smaller cells with a thin cytoplasm. This vegetative cells is the largest cell with a dense cytoplasm, and this with a thin cytoplasm. This is a mature pollen grain. So you can see that in some area of this pollen grain, the enzyme does not grow. This portion is known as the germ pore. This germ pore, it performs some function, but we'll see about that later. So this is a mature pollen grain. It is in a two cell stage. In 60% of the angiosperm, angios the mature pollen grains, it is a two-cell stage. It is a two-cell stage. And then in 40%, it is a three-cell stage. How it has become a three-cell stage? When this pollen grains, when this pollen grain, they falls on a stigma of a female reproductive part, and when it falls, the generative cells undergo another it undergo another mitotic division and give rise into, this is the vegetative and this is the generative, and it give rise into another cells, and here the cytoplasm has become a bit thicker, a bit thicker. Bit thicker. So this is a mature pollen grain, and it concludes the formation of a male gametophyte. So in the next we shall continue with a female